The process of replication in DNA is known as a semi-conservative process. This is because when one double strand of DNA is replicated, each new strand contains one of the old strands of DNA. You can see here the gray bars on top represent the original strand of DNA. When that DNA is split and copied, what we're left with is two new strands of DNA, but each contain one strand from the parent strand. Since each strand of DNA is complementary to the other side, one strand of information can be used to create the opposite strand. This is what leads to the semi-conservative nature of DNA's replication. Here on the bottom right, you can see an image of a strand of chromatin undergoing replication. The arrows indicate what's known as a replication bubble. At certain points in the strand of DNA, an enzyme known as helicase begins to separate one strand from the other. As it does so, the enzyme moves outward and creates a bubble. There's a diagram on the top left that helps illustrate how this bubble forms. As DNA helicase moves outwards, it creates a bigger and bigger bubble. As that bubble gets bigger and bigger, other enzymes replicate the opposite side of the separated DNA. When this process is done, we're left with two full copies of that strand of DNA. Several bubbles can exist on the same strand of DNA at the same time. They eventually all meet and eventually separate to form the two separate strands. Earlier I mentioned that there's a 5' and a 3' end to each strand of DNA. Each side of the double helix has a complementary strand. So while one side is 5' to 3', the other end is 3' to 5'. DNA can only be copied from the 5' end to the 3' end, which means that it must be read from the 3' end to the 5' end. You can see on the top of this replication loop that the arrow is running 5' to 3', but the strand that it's reading runs from 3' to 5'. It's important to note the difference. Now, because we can only copy DNA in one direction, there's a problem on the lagging strand below. How do we copy the DNA on the lagging strand? To do so, the DNA is copied in fragments, and those fragments are later joined. We call those fragments Okazaki fragments. Each one of the arrows on the Okazaki fragments runs from 5' to 3'. Remember, that's the same as on the continuous strand above. It's being copied 5' to 3'. The only difference is that each Okazaki fragment runs into the tail end of the previous Okazaki fragment. After that, those fragments are eventually joined together. This allows genetic information to be copied along each side of the replication bubble. Here you can see a replication bubble with a little more information and a little more detail. Let's unpack this image. Now, we can see the general shape of the replication bubble with a replication fork on each side. In the middle of the forks, we can see the enzyme helicase. That helicase is separating the two strands of DNA from each other and allowing the replication process to occur. The green blocks represent an RNA primer. This is kind of like a starting point for DNA polymerase to begin replicating the strand of DNA. Now, since we're starting from this middle line, the origin, we can see that there's two directions happening, and they're kind of like mirror images of each other. At the top and center left of this replication bubble, we can see that there's an RNA primer. That's at the 5' end of this fragment. Because this fragment running to the left goes from 3' to 5', it can be copied from 5' to 3'. So, DNA polymerase can run from the 5' primer all the way down without any problems. If we move from that point to the right, though, we're going essentially upstream. we're going from the 5' end of this strand towards the 3'. So copying one continuous strand from RNA primer from this point won't work as we go towards the 3' end. The solution is to copy the information through Okazaki fragments. You can see as we go to the right of the origin on the top that there are several segments that go from a 5' end to a 3' end. Those segments stop replicating as soon as they hit the next primer down. Now if we look at the bottom of this replication bubble, we can see that the opposite is happening. And that's because the strand we're copying on the bottom is complementary to the strand that we're copying on top. So everything is kind of reversed. Starting at the origin, going to the left, we can see that going to the left we're going from the 5' end of the strand to the 3' end of the strand. That means that if we are trying to copy to the left, we would be making a copy that is 3' to 5', which doesn't happen. In order to make our copy from 5' to 3', we have to do that in Okazaki fragments. So there are several Okazaki fragments being created as they move to the left. To the right, we're essentially copying downstream. We're making a 5' to 3' copy. So we only have one RNA primer, and then DNA polymerase can copy all the way down the strand. The spots that are filled with RNA primer will later be replaced by DNA nucleotides, and the gaps between the fragments will be sealed by an enzyme called DNA ligase. Here you can see the entire process in summary. On the top, starting from A, we can see that there are several points of replication. In this case, there are 3 different replication bubbles forming. At B, we can see that those replication bubbles have grown as more information has been copied. Within the replication bubbles, there are replication forks at each side. The replication fork is just the point that the genetic information has been separated. At C, we can see that 2 of the replication bubbles have fused. At D, we can see that the replication bubbles have grown. And finally at E, all of the replication bubbles have fused, and now we have 2 separate copies of the same genetic information. The different shades in the lines at letter E are drawn that way to help illustrate the fact that this is a semi-conservative process. Just like those gray bars on the right, each copy contains one strand from the original strand of DNA.